Hello everyone. Just make sure that everything is perfect from my side as well as from yours. Am I audible to you? Good, okay, fine. In today's class, we are going to see some of the MCQs with respect to the cell biology, particularly cellular organelles. Okay? Okay, fine. And for today, your first question is, here's a list of cellular organelles and their function. And after recalling all these questions, I'm going to explain those topics. Okay, those tabular column are very important. They are a consolidation of all the cellular organelles and their function. And your first question is, what is the structure within the nucleus responsible for the synthesis of ribosomal RNA, that is rRNA, and assembly of ribosomes? Okay. Everybody quick. Okay, of course, the site of our ribosome synthesis will be happening only in the place called the nucleolus that is present within the nucleus. And what about this nuclear envelope? Nuclear envelope is made up of how many membrane? This is what my question. Nuclear membrane, nuclear envelope is made up of how many membrane? Two membrane. Okay, and what is that? Outer membrane and the inner membrane, and these two membrane is comprisingly called as the nuclear envelope. And this nuclear envelope will have a lots and lots of pores through which all the materials which has been transferred from the nucleus to the cytoplasm and from the cytoplasm inside the nucleus. Okay, now, and chromatin, we know that will be consisting of the genetic material. And what about this nucleoplasm? What is meant by nucleoplasm? What is nucleoplasm? See, we know that cell, okay, cell has a cytoplasm. Within the cell, we have a cytoplasm through which all the organelles have been present there in the cytoplasm. Okay, like in the same way, we also have a nucleus. Inside the nucleus, there will be a gelatinous substance or gel-like substance, which is called as the nucleoplasm. Very good. Just like a cytoplasm in the nucleus, we are calling them as the nucleoplasm. And your second question, during which phase of the cell cycle does the nucleus undergo a division? Whether it will be the mitosis or meiosis, in which phase of the cell cycle does the nucleus undergo the division? Okay. In cell cycle, whether the nuclear division, that is karyokinesis, happens first or the cytokinesis will be happens first? Always the nuclear division will be happens first, then it will be followed by the cytokinesis, that is cytoplasmic division. For that, this question will be the answer will be the M phase. That is, the nucleus will undergo a nuclear division in the M phase, mitotic phase. And list out the phase which is happening in the mitotic phase. What are all the stages which is happening in the mitotic phase? List out the stages. in the correct order. Okay, that is important. What will be the phase happened in the mitotic division? Prophase, metaphase, anaphase, and telophase. And which two phases are antagonistic to each other? See, you are saying prophase, metaphase, anaphase, and telophase, they are coming under mitotic phase, right? And in which phase they are antagonistic to each other? Obviously, that will be the prophase and the telophase. In prophase, everything will be disappear. Okay, in telophase, everything will be reappear. Okay, this is what happened. And where will be the cytokinesis? That is formation of spindle fibers happens in which phase? Formation of spindle fibers happens in which phase? But the process has been started in the prophase itself. Okay. The spindle fiber starts to form during the prophase of mitosis itself. Okay, don't get confused. Now, which of the following is the site of cellular respiration within the mitochondria? First of all, just tell me what is mean by cellular respiration and what are all the things coming under the cellular respiration? Simply, I'm asking what are all the steps happening in the cellular respiration. After that, answer for this question. Okay, glycolysis, Krebs cycle, that is a tricarboxylic citric acid cycle, or we can also call this the Krebs cycle, and electron transport chain, where the oxidative phosphorylation will happen. Okay, everything will be coming under the cellular respiration. In that, we know glycolysis will be happening in where? Where will be the glycolysis will be taking place? Glycolysis will take place in cytoplasm of the cell. And where will be the Krebs cycle will be happening? Okay. Then where will be the electron transport chain will be happening? Inner mitochondrial membrane. Now the question is, which of the following is the site of the cellular respiration within the mitochondria? For this, you have to keep in mind where the Krebs cycle is going to happen, because in glycolysis we are going to synthesize a minimum amount of ATP, but in case of this kindly switch off your video please. See, in glycolysis we are going to synthesize a very less number of ATP when compared to the Krebs cycle and mitochondria. Okay, so whenever they are asking a question like this, you should not consider about this electron transport chain, but instead of that, you have to keep in mind with the TCA cycle. And now tell me the answer. What will be the site of this particular cellular respiration within the mitochondria, where it is going to happening? The Krebs cycle will be happening in the matrix of the mitochondria. There is no point of having a confusion. Someone has given me the answer as crystate. Okay, it is no way it is a crystate. Okay, now see, matrix will be the innermost compartment of the mitochondria. We know that, just like a cytoplasm. Okay, and see where the citric acid cycle or the Krebs cycle and the fatty acid oxidation will be happening. Okay, now, okay, just tell me, you are saying that glycolysis, Krebs cycle, all other things are happening under the cellular respiration. How many number of ATP is going to synthesize in this cellular respiration? Okay, and what will be the gross ATP? Whenever I'm asking how many number of ATP, you should always consider about the gross. Okay, gross will be 38. Total ATP production in the cellular respiration will be the 38. And in case of net, you have to say 36, and 2 will be used in the synthesis process. Okay, now, which of the following metabolic process occurs in mitochondria? Photosynthesis, glycolysis, beta oxidation, and transcription. Very good, beta oxidation. Generally, what will be the functions of this mitochondria? Okay, see, oxidative phosphorylation, electron transport, and ATP synthesis will be happening in the mitochondria, as well as at the site of synthesis of amino acids and some of the heme groups. Okay, and also the regulation of calcium concentration. Okay, also apoptotic events, certain apoptotic events, all these processes will be happening in the mitochondria. Okay, okay, the next question will be, which cellular process involves the Golgi apparatus in the secretion of cellular products? Endocytosis, exocytosis, phagocytosis, and pinocytosis. What will be the name of the process? It will be the exocytosis. Why? Because it is the one, see, in the nucleus, all the important proteins has been transported to endoplasmic reticulum. In endoplasmic reticulum, we have two types of endoplasmic reticulum. One is rough and smooth endoplasmic reticulum. Rough is meant for the synthesis of proteins and smooth is meant for the synthesis of lipids. Okay, after the synthesis of proteins or lipids, it has to send to the Golgi complex. Okay, there are some of the important COP proteins are there. Okay, COP2, COP3, and COP1 and proteins and clathrin protein are also involved here. Okay, through this transformation. And after this, this Golgi, from this Golgi complex, the important protein in the form of vesicles, they have to ship to some of the important organs like lysosomes, plasma membranes, or even outside the cell. Okay, everything will be happening only from the Golgi apparatus. It is also known for its shipping and packaging of the cell. Okay, now for that, this process is said to be as the exocytosis process. Okay, now, which of the following conditions can result from malfunctioning of the lysosomes? See, if some of the enzymes has been missed from the lysosomes, it leads to a certain conditions. And what will be the name of the disease if the lysosome is not working well? Can anyone say what will be the symptom of Tay-Sachs disease? And what will be the pH maintained inside the lysosome? Whether it will be the acidic pH or alkaline pH? Very good. That will be an acidic pH where we can find many of the hydrolytic enzymes. Because of the pH maintained inside the lysosome, we are calling this lysosome as the suicidal bags, where we can find the unwanted materials are going to digested inside the lysosomes. When it comes to the Tay-Sachs disease, it is somewhat a deficiency of one of the enzyme inside the lysosome, and that enzyme is responsible for breaking down of a certain types of fat. Okay, and that enzyme is called as the hexaminidase A. Okay, so because of the absence of this enzyme, it leads to a disease called Tay-Sachs disease. Very good. Nerves degeneration. Now, which organelle is responsible for modifying and packaging protein synthesized by the rough endoplasmic reticulum? Just now I have explained you. Obviously, that will be the Golgi complex. I don't want to explain this. And which of the following process occurs within the endoplasmic reticulum? Glycolysis, Krebs cycle, protein folding, photosynthesis. What is the process occurs within the endoplasmic reticulum? Shall we say this as a post-translational modification? Yes. Okay. See, protein folding, modification, and sorting of proteins occurs within the endoplasmic reticulum. That too, particularly the lumen of the rough endoplasmic reticulum. Lumen is nothing but the space present inside the rough endoplasmic reticulum. And what will be the difference between the rough and the smooth endoplasmic reticulum? This is the basic difference. If the endoplasmic reticulum is attached with the or embedded with the ribosomes, then we are calling them as the rough endoplasmic reticulum, and because of the ribosome present in the endoplasmic reticulum, it is going to dealing with the synthesis of proteins. Right, and what is the primary function of peroxisome in the cell? Detoxification of harmful substances. Name the important enzyme present inside the peroxisome which is involved in the detoxification process. Name the important enzyme. Name the important enzyme that is responsible for this destroying all the harmful substances present inside the peroxisome. Hello, I'm asking you, do you know or not? The important enzyme responsible for all these destroying or degrading this toxic materials inside the peroxisome. And of course, very good catalase and other important enzyme, peroxidase enzyme. Okay. Okay. Do peroxisome have a DNA? Do peroxisome have a DNA? Okay. Do peroxisome has a ribosome? Do ribosome has a peroxisome? Sorry, do ribosome is present in the peroxisome? Obviously, no. Okay. So do peroxisome involved in the oxidation of involved in the beta oxidation? Do peroxisome involved in the beta oxidation process? Okay. Mitochondria is also involved in the beta oxidation process, and peroxisome is also involved in the beta oxidation process, but which will be the most important organelle involved in the very long chain of fatty beta oxidation of very long chain of fatty acids? See, what I'm asking is mitochondria and peroxisome, both organelle are involved in the beta oxidation process. Beta oxidation of fatty acids through which we can generate an ATP. But which organelle is responsible for beta oxidation of very long chain of fatty acid? Very good. That will be the peroxisome. Of course, mitochondria is also involved in such process, but it is dealing with a beta oxidation of very small chain of fatty acids. Okay. Now, next question. Which of the following molecules is synthesized in the peroxisome? Okay, see, it has no DNA, it has no ribosomes to synthesize proteins. Okay, just now we have studied that it is involved in the synthesis of certain lipids, include the oxidation of fatty acids, okay, which helps in the breakdown of long chain fatty acids. As a result of breaking down of fatty acids, what we are going to do? What we are going to get by oxidizing the fatty acids? Generally, ATP protection is involved only in case of ATP protection. It is involved only in mitochondria, but not in the peroxisome. So, what will be produced in case of peroxisome? That will be the fatty acids. Okay. Now, what is the structure within the peroxisome where fatty acids are oxidized to produce acetyl-CoA? What will be that? That will be obviously the peroxisomal matrix. Okay. Now, see, all the fatty acids, these fatty acids are getting oxidized only in the peroxisomal matrix. Because why it is happening in the peroxisomal matrix is because all the required enzymes is present only in the matrix of the peroxisome. See, now these fatty acids have been broken into what? Acetyl-CoA molecules, not as ATP. Okay, ATP protection is just confined only to the mitochondria, right? Now, which of the following condition is associated with the dysfunction or deficiency of protein? Sorry, peroxisome. Alzheimer's disease, Tay-Sachs disease, Zellweger disease, sickle cell anemia. Zellweger disease, of course it is right. Is it an inherited disease? Is it an inherited disease? Is it an inherited disease? Of course, it will be the rare inherited disease. Okay. See, in peroxisome, what is the condition of this Zellweger syndrome means it looks like an empty membranous ghost. Okay, which means what? It lacks such enzymes. Okay, so if it lacks such enzyme, the peroxisome they are failing to import some enzymes from the cytosol to the peroxisome. Okay, in case of absence of that particular enzyme, the peroxisome can't import some of the enzymes from the cytoplasm to the mitochondria to continue its activities. So such condition which leads to the Zellweger syndrome, right? See, it is a rare genetic disorder that will be characterized by the absence or malfunctioning of the peroxisomes. It leads to the neurological abnormalities. And which plastid is specialized for the storage of starch in plant cells? Okay. Do plastids have a DNA? Do plastids have a DNA? Okay. And how many types are there in plastids? Generally, we can classify this plastid into two categories. Very good. Chromoplast and leucoplast. And this chromoplast is further categorized into chloroplast, rhodoplast, and pheoplast. Okay. And leucoplast will be further categorized into amyloplast, elaioplast, and proteinoplast. Okay. See, what is the difference between this chromoplast and the leucoplast? This is the two types of plastids. Generally, this chromoplast, it is calling them, we are calling them as the colored plastids. But in case of leucoplasts, we are calling them as the colorless. Okay, based on a pigment, they have been categorized into two. And if it has a pigment, see, chromosome has a pigment. Okay, leucoplasts, they are not having a pigment. As it have the pigment, these chromoplasts are involved in the function of synthesis. Very good, synthesis function. In case of leucoplast, as it lacks the pigment, it can't synthesize anything, but it has the capacity to store something. Okay. In storage, they are classified into leucoplast will be further categorized into amyloplast and elaioplast and proteinoplast. And this amyloplast is the one going to store amylose, that is sugar, in tubers, cotyledons and endosperms, like that. Okay. And in elaioplast, they are the one going to store oil. And in proteinoplast, they are going to store protein. Okay, in chromoplast, here comes the chloroplast, rhodoplast, and pheoplast. We know about what is chloroplast. And in a rhodoplast, they are act as a gravity. And this pheoplast is the one this is acting as the, it is somewhat related to the plants. Okay, these are all the types. This flowchart is very important. Okay, have a note of it. Now, the next question. Which plastid is responsible for the synthesis and storage of pigments other than chlorophyll, such as carotenoids and anthocyanin? It is responsible for the synthesis and storage of pigments other than the chlorophyll, such as carotenoids and anthocyanins. See, that will be the chromoplast. This is the one responsible for the synthesis and storage of a pigment other than the chlorophyll. This is the one gives rise to a diverse colors. This is the one responsible for giving colors to parts of the plants. And what is the name of the process by which the vacuoles fuse with the lysosome to degrade cellular waste and maintain cellular homeostasis? This is somewhat related to the organelle called the vacuole. I just answered to my question. This vacuole is present in the plant cell or animal cell? It is restricted to the plant cell. Now answer to this question. What is the name of the process through which this vacuole is going to fuse with the lysosome and they are going to degrade the waste materials? Okay, by degrading all these waste materials, they are maintaining the homeostasis of that particular organism. Here, this will be the autophagy. The autophagy is the process through which the vacuole is going to fuse with the lysosome and by degrading the all, and it leads to the degradation of all the waste materials. Okay, now, which of the following is the primary component of the cell wall in plants? That will be the primary component which is make up the cell wall of the plants. To cell wall present in plant cell or animal cell? Or it can be present in both the cell? The cell wall present in, of course, it is present only in the plant cell. It is not present in the animal cell. And that cell wall is made up of a cellulose. That is why plant is very hard to cut. And the cellulose is made up of cellulose is made up of cellulose is made up of. Do you heard about hemicellulose? This is the most strongest supportive system in the plant cell. Okay, and what is their function mainly? They are giving a skeletal system to the plant. Okay, and they are the one determining the size and shape of the cell, and they are also the one going to determine the limits of expansion and water uptake. Okay, how it has to enter into the plant like that. Okay, see, when compared to the plasma membrane or the cell membrane, which is the most active part of the cell wall is the most active part of the cell. Okay, like we know that plasma membrane have a lots and lots of receptors in it. Like in the same way, cell wall also have many receptors. So cell wall is a living part of the cell or not? It is a living. It is a living part of the cell and it receives a signals from the hormones. It does have a receptors in it. Okay, and what is their composition? Generally, cell wall is made up of two portions. One is a fibrous and non-fibrous portion, and in fibrous, see, generally cell wall it has a two portion. Okay, one is a fibrous portion and another one is non-fibrous portion. Okay, in fibrous portion it is made up of a cellulose, hemicellulose, microfibrils. Okay, hemicellulose, microfibrils. And in non-fibrous portion it is made up of a glycoprotein and the pectin. Okay, these are all the protein and the carbohydrates which make up the non-fibrous portion of the cell wall. Okay, and we know that this cellulose is always cross-linked with the hemicellulose microfibrils and they are tightly packed with this glycoprotein and pectin. Okay, see, generally this cell wall is divided into two portions, fibrous and non-fibrous portion. This fibrous portion is made up of a cellulose and hemicellulose, and this non-fibrous portion is made up of a glycoprotein and pectin. See, this cellulose is tightly packed with this hemicellulose. Okay, they are cross-linked with this hemicellulose. How they are packed? They are packed with this non-fibrous portion, that is glycoprotein and pectin. Okay, clear? Now, next question. In addition to the cellulose, which other polysaccharide may be found in the cell walls of some plants, providing additional strength and water resistance? Starch, hemicellulose, lignin, and pectin. Is it a pectin? See, as I have said before, cellulose is the most strongest supportive system. After cellulose, lignin is considered as the most strongest protein. Okay, don't get confused. So the answer will be the lignin. Now, which of the following cellular process involves the cytoskeleton? For which of these process they requires a cytoskeleton, whether it will be the cellular respiration, cell division, DNA replication, and protein synthesis. Okay, cell division, particularly in which phase? Particularly which phase? And we have studied there are different, okay, is it not in metaphase? Okay, it is started in the metaphase. Anaphase also, the cell cytoskeleton plays a crucial role. Okay, now, okay, let me ask you a simple question. And in this cytoskeleton, we have different types of protein: microtubule, microfilament, and intermediate filaments. Of these things, spindle fibers is given by synthesized by which of the following cytoskeletal proteins? Spindle is formed by, it is assembled by, which is the one going to assemble as a spindle? We can say like that. Microtubule or microfilament or intermediate filaments. Very good. Microtubule is the one assembled into a spindle fibers. Which organelle is responsible for organizing and anchoring microtubules in animal cells during cell division? The main organizer of this particular cytoskeleton will be the centrosome, okay, through which it can be attached. Which type of proteins span the entire lipid bilayer of the plasma membrane and are involved in the transport of molecules across the membrane? Okay, okay. We have peripheral proteins, we have an integral proteins, and we have a transmembrane proteins. Okay, in the plasma membrane, we know that. And which protein it is easy to separate from the plasma membrane just by adding some detergent? Peripheral proteins, because it is attached in the surface itself. In the integral membrane or the transmembrane integral protein or the transmembrane protein, it is embedded within the membrane. Okay, okay, let me say you a statement that you have to tell me whether it is right or not. All transmembrane proteins are integral, but not all integral proteins are transmembrane proteins. This is what my statement. I repeat, all transmembrane proteins are integral, but not all integral or the transmembrane protein. Whether the statement is right or wrong. See, integral protein, it is an umbrella term. Okay, it is integral protein, and the transmembrane protein will be the type of the integral protein. So we can say all transmembrane protein are the integral protein, but not all integral protein are the transmembrane protein. See, I repeat the statement. Transmembrane protein. Okay, transmembrane protein will be the type of integral protein. Okay, integral protein has a type called the transmembrane protein. So all the transmembrane protein are definitely has to be present. All transmembrane proteins or integral protein. So the statement is a right statement. I'll repeat the statement, just make a note of it. All transmembrane proteins are integral, but not all integral proteins are transmembrane protein. Now, what is the difference between these two proteins? These integral proteins are embedded inside the membrane, and transmembrane protein is also going to embedded inside the membrane, but it is moving along the plasma membrane. Okay. Now, which of the following molecule helps stabilize the plasma membrane and maintain its fluidity at different temperatures? Very good. Cholesterol will be the correct answer. Okay. Okay. We have a saturated fatty acids and unsaturated fatty acids, right? In saturated fatty, and we have two types of fatty acid, and which type of fatty acid, whether it is a saturated or unsaturated fatty acids, or dealing with the rigidity, which is responsible for the rigidity of the membrane. Which is responsible for rigidity. Very good. Saturated fatty acids is responsible for the rigidity because the saturated fatty acid is solid at room temperature. Okay, it is solid at room temperature. It can't metabolize easily. But in case of unsaturated fatty acid, it is responsible for its fluidity. Okay, it is liquid at the room temperature. For example, all our vegetable oils are coming under the unsaturated fatty acids. In which type of fatty acids we can find the double or triple bond? In which fatty acids we can find the double or the triple bond? Very good. Unsaturated fatty acids has the double or the triple bond. Okay. If we have one double bond, we are calling them as the MUFA, mono unsaturated fatty acid. If we have one double bond, we can call them as the MUFA, mono unsaturated fatty acids. If we have more double bond, okay, more double bond in the fatty acids, we can call them as the polyunsaturated fatty acids. Okay, if the MUFA and the PUFA are high, if it is that we know that unsaturated fatty acids have the double or triple bond, and mono or PUFA are definitely they will be the unsaturated fatty acids. More amount of MUFA and PUFA, the oil will be healthy or not. This is what my statement. More amount of MUFA or PUFA, the oil will be healthy or not. It will be healthy. Okay. See, there are a lot to discuss about this plasma membrane. Okay. And also, what will be the name of the model? It is going to explain the structure of plasma membrane. Name the model. Name the model. Just put forth two, which is explaining the, yeah, Singer and Nicholson will be the name of the scientist. Yeah, of course, 1972. Yeah, the name of the model will be the fluid mosaic model. Okay. Good, okay. Okay, in membrane, which constitutes a major part, whether it will be the lipid or protein or carbohydrate. In a plasma membrane, which secretes a major portion. Lipid will be the major portion, and after that followed by the Yeah, of course. Lipid will be lipid followed by the protein, and it is followed by the carbohydrates. Right? Okay. And you should know whether it will be the it has a lipid bilayer, right? Icebergs on the sea. It is what the model suggests. It is a lipid bilayer, and it has a non-polar region and polar region. And generally, this non-polar region will be the tail region, that is hydrophilic. It is the one facing the core. So all these structures are very important. Kindly have a look at that. Okay. That's all about the questions. Now I have attached some of the important tables. You have to read that. Okay. For the first table will dealing with the nucleus. Okay. This is what short. See this nucleus, mitochondria, endoplasmic reticulum, Golgi complex. These are all the list of cellular organelles and their functions. And majorly for plasma membrane, I have inserted this. See how it will be looks like. It will be look like a phospholipid bilayer, and in this layer we are having a lipid, protein, cholesterol, and the glycolipids. Okay, these are all the important structure which make up the plasma membrane, and their compositions will be, we know that hydrophilic, hydrophobic. Okay, always hydrophilic will be the head. Hydrophilic means what? Water loving. So it will be facing outwards, and the hydrophilic tail will be the facing inward. Okay, and fluidity, and which fatty acid is responsible for fluidity, which is responsible for the rigidity. All these things are very important. We know that it is selectively permeable membrane, and also we have a transport mechanisms are there. Okay, in plasma membrane we are having a different transporters, different receptors, because one cell, it has to be communicate with the other cell only with the means of this plasma membrane. So all the important cell communication and cell signaling, important proteins, everything will be present in the plasma membrane. And in the passive transport will also be happening, active transport will also be happening. These are all the types of passive transport, where we can find no energy is energy is required for this type of transport, diffusion and facilitated diffusion. In case of active transport, it needs energy to transfer from the to take inside the cell or even from the outside the cell. And cell signaling, cell adhesion for protection, recognize something, and to maintain this shape, as cell wall is not present in the animal cell, what is the one responsible for maintaining the shape in case of animal cell? Obviously that will be the plasma membrane. And when it comes to the nucleus, we know that you have to understand what are all the process. See, we know that replication, transcription, and translation. Replication and transcription will be happening in the nucleus, and translation will be happen in the cytoplasm. So all these things are very, very important. And what type of RNA is synthesizing in the nucleus, everything will be an important phenomena. And what is the important protein will be present in the I forgot to say this. There are 3 different types of transporters present in the plasma membrane. Okay, flippase, floppase, and scramblase. Okay, and you should know that flippase will be ATP dependent or ATP independent. It can work either way. But floppase, it is ATP dependent. Okay, in scramblase, it is calcium dependent. All these things are also important. You have to read about the transporters. That is very important to read. I also read about the chloroplast. That is very, very important. When you read about the photosynthesis, you'll cover the chloroplast. And one more question is, okay. In signaling molecule, okay, we'll deal this signaling molecule tomorrow. Okay, for today's class, this cellular organelles is enough. Okay, for tomorrow, what you're going to read, and from that topic, I'll take some of the questions. We have studied today questions from the cellular organelles, right? And if you have time, kindly revise the plasma membrane as well as the what are all the topics we have took today. Okay, cell organelles and important functions with respect to the plasma membrane. We have covered today these two units, these two subtopics. Tomorrow we are going to discuss the organization of genes and chromosomes. Okay, just read this, and also cell division and cell cycle. For tomorrow's topic, you have to read these two types, these two subtopics. Clear? Any doubts? Okay. If you have any doubts, you can stay. Otherwise, you can leave the class. Tomorrow, the same time, 8:30 p.m. For tomorrow, read the cell division and organization of genes and chromosomes. These two are the discussion for tomorrow. Okay. Okay. Thank you. Revise all these things.